Hello everyone, welcome back to CAPSA. In this lecture, we are going to explore the basic components, structures, and the function of the immune system. So stay with me till the end of this video because each section builds on the next, and by the end or the conclusion, you will have a strong foundation in the immunology, which is perfect for the both exam and the real life understanding. The immune system is nature's defense force, carefully designed to protect us from the infections. So, without it, the survival would be impossible. Individuals with weak or absent immune responses often succumb to infections very early in their life. Our immunity operates at two levels. First, we have innate immunity, which is immediate but less specific. Then comes adaptive immunity, which is slower to respond initially, but it is highly specific and develops the memory. Let's break down the two main arms of the immune system: innate immunity, which recognizes general patterns found in the microbes. These are called PAMPs, or the Pathogen-Associated Molecular Patterns. It uses receptors encoded in our genes known as pattern recognition receptors. The response is immediate, but it does not form memory. Adaptive immunity, in contrast, recognizes very specific molecules thanks to the receptors generated through genetic rearrangements. These include T cell receptors, B cells receptors. The response takes longer because cells need to interact, but once activated, adaptive immunity forms the memory, which is why vaccine works. Innate immunity uses players like dendritic cells, macrophages, neutrophils, and natural killer cells. Adaptive immunity involves B lymphocytes that makes antibody and the T lymphocytes that carry out cellular responses. In short, innate is fast but non-specific, while adaptive immunity is slow but specific and long-lasting. Now let's highlight some of the important stars of the immunology. Antigens, which are substances recognized as a foreign and capable of provoking an immune response. Now, what is antibodies? These are immunoglobulins, or the proteins produced by B cells, and they are highly specific and act as the weapons of the adaptive immune system. T cell receptors recognize the fragments of antigens, but only when presented by specialized molecules called the major histocompatibility complex, or MHC. MHC class 1 represents two CD8 T cells, and MHC class 2 represents the CD4 T cells. Alongside this, we also have accessory molecules that enhance communication and binding between the immune cells, making the response much more efficient. How does the immune system actually recognize invaders and get activated? Immune, innate immune cells use pattern recognition receptors, such as toll like receptors, to detect the common microbial structures. This triggers the initial alarm system. For adaptive immunity, antigen must first be processed and displaced on MHC molecules. This process is called antigen presentation and is essential for T cells recognition. But recognition alone is not enough. T cells also need co stimulation through accessory molecules. Without this second signal, T cells remain inactive, preventing unnecessary or accidental activation. This dual signal requirement ensure precision in the immune response. Once activated, how does the immune system actually fight? There are two main branches: humoral and cellular. Humoral effectors include antibodies and the complement system. Antibodies neutralize the pathogens, tag them for detection, and activate complement proteins, which punch holes in the microbial membranes. Cellular effectors include phagocytes such as neutrophils and macrophages that engulf pathogens, and cytotoxic lymphocytes like CD8 T cells, NK cells, which directly kill infected cells using toxic proteins like perforin and granzymes. Finally, we have cytokines and chemokines, the messengers of the immune system. Cytokines coordinate the immune attack, while chemokines guide immune cells to the right place at the right time. Antibodies are fascinating molecules. Structurally, they are made of two heavy chains and the two light chains, with variable regions for antigen binding and contrast regions for effector function. Different antibody classes or isotypes serve different purposes. IgM is the fast responder and an excellent activator of the complement. IgG is the most abundant in serum, can cross the placenta and enhance the phagocytosis. IgA protects the mucosal surfaces like the gut and the respiratory tract. IgD is mostly a receptor on B cells. IgE plays a key role in allergy and defense against parasites. Each class of antibody is specialized, ensuring our immune system can tackle pathogens in every environment of the body. One of the most remarkable features of the immune system is its diversity. How does the body generate millions of different receptors from a limited number of genes? The answer lies in genetic recombination. B and T cell receptors are created by rearranging V, D, and J segments in different combinations. Beyond this, somatic hypermutation introduces additional changes, allowing antibodies to fine tune their binding to antigens. This mechanism gives our immune system an almost limitless ability to recognize new pathogens, a crucial advantage in an ever changing microbial world. And with that, we have completed our introduction to immunology and the structure and the components. The immune system is not just about defense, it's a beautifully coordinated network of recognition, communication, and the memory. So if you have found this lecture so much helpful, make sure to like, share and subscribe to CAPSA. Don't forget to hit the bell icon so you don't miss our upcoming lectures where we will dive even deeper into the immunology. Thank you for staying till the end. Your learning journey is just the beginning.